So let's shift gears a bit now
Okay.
and dive into the world of chemical bonds and molecular structure.
Let's do it.
Understanding how atoms bond together
Mm-hmm.
and how those bonds create the 3D shapes of molecules.
Yes.
It's crucial for understanding how all these biomolecules actually work.
Absolutely. It's all connected.
Okay, so let's start with a quick review of the periodic table.
The periodic table.
I know it can seem overwhelming at first glance,
It can be a bit intimidating.
but it's actually a beautifully organized system.
It is.
It tells us so much about the elements.
It's our roadmap to the elements.
I like that.
Hey.
And remember, when it comes to our bodies, just 4 elements make up over 99% of the atoms,
That's right. Hydrogen, oxygen, carbon, and nitrogen.
the building blocks of life.
Exactly.
Hydrogen and oxygen make water, of course.
Of course.
But carbon is the real star when it comes to building those complex biomolecules.
It is.
It has that amazing ability to form 4 stable bonds
Mm-hmm.
because of orbital hybridization.
Exactly. Carbon's electrons, they can rearrange themselves into these hybrid orbitals that are perfectly positioned to form strong bonds with other atoms.
So it's like carbon has these versatile connector pieces
I like that analogy.
that let it create so many different molecular structures.
It's like a molecular Lego set.
I love that.
Yeah.
So these hybrid orbitals allow carbon to form single bonds, like in methane,
Right.
but also double bonds, like in carbon dioxide,
Yep.
and even triple bonds, like in acetylene.
Exactly. And those double and triple bonds, they're important in a lot of biomolecules,
Right.
giving them their unique shapes and properties.
Okay, now let's talk about the different types of chemical bonds that hold these biomolecules together.
All right.
I remember learning about covalent bonds, ionic bonds, hydrogen bonds,
It's classic.
but it's all a bit fuzzy now.
Let's refresh your memory then.
Yeah.
Covalent bonds, those are the strongest.
Okay.
Atoms share electrons in those bonds.
Mhm.
Think of them like the glue holding most molecules together.
Okay, so those are the strong ones.
Yep. Then we have ionic bonds,
Right,
where oppositely charged ions attract each other.
like it's salts.
Exactly. And finally, hydrogen bonds,
Okay.
weaker than covalent or ionic bonds,
Right.
but super important in biomolecules,
They are?
like holding those 2 strands of DNA together.
It's like a hierarchy of bond strengths, each with its own special role
Absolutely.
in building and stabilizing those complex biomolecules.
Right. Now let's zoom in a bit closer
Okay.
and talk
Okay.
about molecular structure.
Okay.
We often use those simple 2D formulas to represent molecules,
Right,
but those are just simplified.
right. They're not the whole picture.
No, because molecules actually exist in 3 dimensions,
Right.
and their shapes are key to how they function.
Okay.
Remember those models from chemistry class? The ball and stick models, the space-filling models?
Yeah, I do.
Those really helped visualize how atoms are arranged in space.
They did.
And understanding those 3D shapes, it's critical for understanding how molecules interact with each other.
Right.
Think about enzymes, for example.
Right.
They have those specific active sites
Mhm.
shaped to perfectly fit their substrates.
Like a key fitting into a lock.
Exactly. So the shape of a molecule,
Mhm.
it's just as important as its chemical composition.
When it comes to its activity.
You got it. And that brings us to bond polarity.
Oh, yeah. Bond polarity.
That's the unequal sharing of electrons in a covalent bond.
Right. Some atoms, like oxygen, are more electronegative.
Exactly. They pull electrons closer,
Yeah.
creating a dipole moment.
Like we see in water.
Yes. The oxygen atom is slightly negative.
And the hydrogens are slightly positive.
You got it. And those dipole moments,
Mhm.
that's what lets water molecules form those hydrogen bonds.
With each other
Yeah.
and other polar molecules.
Exactly. Now let's talk about isomers.
Okay, isomers. Those are molecules with the same chemical formula, but different arrangements of atoms in space, right?
Right.
I always found those mirror image molecules so fascinating, and enantiomers.
Yeah, and enantiomers, they have the same atoms and bonds,
Mm-hmm.
but they're arranged differently in 3D space.
Right.
And that small difference can have huge consequences,
Yeah.
especially in biological systems.
You can.
Often, only one enantiomer of a drug will work,
Right.
while the other might be totally useless
Wow.
or even harmful.
That's crazy. Such small differences can have such big effects.
They can. Now, we've been talking a lot about structure,
Yeah.
but let's not forget about energy.
Okay.
Thermodynamics is key for understanding how biological systems work.
Okay.
Remember those terms? Enthalpy, entropy, and Gibbs free energy?
I do. But they always felt a bit abstract.
Let's make them more concrete then.
Okay, let's do it.
So enthalpy, we usually use H for that.
Okay.
It's a measure of the total energy of a system.
Like the heat content.
Yeah, think of it like that.
Okay.
Entropy, that's S, that's all about disorder or randomness.
Okay.
So a system with high enthalpy, it's got lots of stored energy.
Right.
And one with high entropy, it's very disordered.
Like my desk at the end of a long week.
Perfect analogy.
So how do these concepts apply to biochemistry?
That's where Gibbs free energy comes in, G.
Okay.
It combines enthalpy and entropy into
Nice.
one value that tells us if a reaction will happen spontaneously or not.
So if the Gibbs free energy change, the delta G, is negative, it means the reaction will happen on its own.
Exactly. That's a spontaneous reaction.
Okay.
Or exergonic. If it's positive, it's non-spontaneous. Or endergonic, meaning it needs energy input to happen.
Got it. What about enzymes? They're catalysts, they speed things up, but do they change Gibbs free energy?
Great question. They don't actually change the overall Gibbs free energy change.
So they don't create energy?
No magic there. They lower the activation energy instead.
Okay.
That's the energy barrier that reactants have to overcome to become products.
So they make it easier for the reaction to start.
Exactly. They provide a different path for the reaction that needs less energy to get going.
So without enzymes, lots of biochemical reactions would be way too slow.
Way too slow for life.
Now, what about those group transfer reactions that are so common in metabolism? I remember learning about ATP, the energy currency of the cell.
Right, ATP.
I could use a refresher on how that all works.
Think of group transfer reactions like exchanging currency.
Okay.
Chemical groups, like phosphate groups, are moved from one molecule to another.
Right.
ATP, adenosine triphosphate, is the perfect example.
Okay.
It's got 3 phosphate groups linked together by those high-energy bonds.
Right.
And when one of those phosphate groups is transferred,
Yeah.
it's like spending energy.
Right.
That transfer is often paired with an endergonic reaction, providing the energy it needs to get going.
So ATP is like a rechargeable battery.
That's a great analogy.
Yeah.
Storing energy in those phosphate bonds
Okay.
and releasing it when needed.
And this is happening all the time in our cells.
It is.
Powering everything.
Muscles, protein synthesis, you name it.
Now, what about those coupled reactions you mentioned? How do those work exactly?
It's like a 2-for-1 deal, biochemically speaking. You've got an endergonic reaction that wouldn't happen spontaneously,
Right.
but it's linked to a very exergonic reaction like ATP hydrolysis.
So the energy released from that exergonic reaction is used to power that endergonic one.
Exactly, like using a waterfall to make electricity.
That's a good one. And this coupled reaction thing, it's so important for understanding how our bodies use energy.
It's fundamental. We'll see it again and again when we look closer at metabolism.
Okay, before we get to metabolism, let's do a quick review of those biomolecules
Okay.
we've been talking about.
Get building blocks.
Yeah, the 4 major classes: carbs, lipids, proteins, and nucleic acids.
All right. Carbohydrates, they're sugars and starches, our main energy source.
Right. Then lipids, those are fats, oils, and waxes.
Important for storing energy, insulation, and cell signaling.
And proteins, the workhorses of the cell.
Doing it all. Catalyzing reactions, transporting molecules, and more.
And finally, nucleic acids, like DNA and RNA.
Storing and transmitting that genetic info.
Okay, I think we've built a good foundation here.
I think so too.
Now it's time to tackle the metabolic maze. But before we get lost in the pathways, can you remind us what exactly metabolism is all about?
So metabolism, it's like the sum of all the chemical reactions happening inside a living organism to keep it alive.
Okay.
It involves both breaking down molecules to get energy.
Right.
That's catabolism.
Okay.
And building molecules up, which needs energy.
Right.
And that's anabolism.
It's a constant cycle.
Exactly, like a dance between breaking down and building up.
Okay, so what are some of the key metabolic pathways we'll be exploring in this deep dive?
Well, some of the most important ones are glycolysis,
Okay.
the citric acid cycle, also known as the Krebs cycle,
Right.
oxidative phosphorylation, fatty acid oxidation, and amino acid metabolism.
Okay, let's start with glycolysis, that classic pathway that breaks down glucose for energy.
Yes, glycolysis.
I remember it happens in the cytoplasm and doesn't need oxygen.
That's right. It's an anaerobic pathway.
Okay.
It converts one glucose molecule into 2 pyruvate molecules
Okay.
and makes a bit of ATP and NADH in the process.
And ADH, that's an electron carrier.
Yeah.
That's important for oxidative phosphorylation, right?
You got it. Now, what happens to pyruvate depends on whether there's oxygen around.
Okay.
If there is aerobic conditions, pyruvate goes into the mitochondria,
the powerhouses of the cell,
That's right.
where it's oxidized further in the citric acid cycle.
Exactly.
Ah, the citric acid cycle. I remember that it's a cycle
It is.
that makes a lot of NADH and FADH2 another electron carrier.
Precisely. And those electron carriers, they're used in oxidative phosphorylation,
Okay.
the last step of cellular respiration to make most of the ATP.
Right, oxidative phosphorylation.
Happens in the inner mitochondrial membrane
Yes.
and uses those electron transport chains and ATP synthase.
It's a pretty remarkable process.
It is. It uses the flow of electrons to create a proton gradient across that membrane, and then ATP synthase uses that gradient to make ATP.
Amazing. Okay, so we've covered the main pathways for glucose. What about fatty acid oxidation? How do we break down fats for energy?
So fatty acid oxidation, also called beta oxidation, happens in the mitochondria.
Okay.
And it involves a series of steps that remove 2 carbon units from the fatty acid chain,
Okay.
producing acetyl-CoA, NADH, and FADH2.
And that acetyl-CoA can then go into the citric acid cycle.
It can. So both glucose and fatty acids
Right.
can fuel that cycle
Right.
and make ATP through oxidative phosphorylation.
Okay, what about amino acid metabolism? I know proteins can be broken down for energy, but they also have other jobs in the body.
They do. They're very versatile.
But how does that work?
Well, amino acids can be used for energy,
Okay.
but they're also essential building blocks for proteins and other biomolecules.
Right, right.
Amino acid metabolism involves a whole network of pathways that break down amino acids, make new ones, and convert them into other things like glucose or ketone bodies.
Ketone bodies, those are made during fasting.
Yeah.
Or in certain conditions like diabetic ketoacidosis, right?
Exactly. When there's not enough glucose around, the body can switch to using ketone bodies,
Right.
which are made from breaking down fatty acids
Okay.
as an alternate fuel source.
This is all making sense now. It's amazing how interconnected these pathways are, always adapting to what the body needs.
It's a beautiful system.
It is. But how is all this metabolic activity controlled? It can't just be a free-for-all, can it?
No, it's tightly regulated.
It has to be.
Metabolic regulation is key for maintaining homeostasis
Right.
and making sure our cells are getting what they need when they need it.
Okay, so what are the main ways this regulation happens?
Metabolic pathways are regulated at different levels,
Okay.
including allosteric regulation of enzymes, hormonal control, and changes in gene expression.
Allosteric regulation. That's like those dimmer switches we talked about.
It is, adjusting enzyme activity by binding those effector molecules.
And hormonal control plays a big role.
It does. It coordinates metabolic activity in different tissues.
Give us an example.
Sure, like insulin,
Like,
made by the pancreas, it helps cells take up and store glucose.
okay.
While glucagon, another hormone from the pancreas, tells the body to release glucose from storage.
So it's like a hormonal orchestra.
I like that. Different hormones fine-tuning the metabolic activity.
And then changes in gene expression
Yes.
can change the levels of enzymes,
And other proteins involved in metabolism.
providing a long-term way to adapt.
Exactly. So it's a multi-layered system of control,
It is.
making sure our metabolism is finely tuned to meet our needs.
I think it's time for another round of questions.
You think so? All right, let's do it.
Are you ready to test our listeners again?
Fire away!
All right, let's dive into those questions then.
Okay, first one. How does that dipolar nature of water make it such a great solvent? This one is key.
It is a big one.
So water's dipolar nature, it comes from how the electrons are spread out between the oxygen and hydrogen atoms.
Right.
The oxygen atom, it's more electronegative, meaning it pulls electrons closer,
Okay.
so it gets a partial negative charge.
Right.
And the hydrogens, they get a partial positive charge.
Okay.
And those partial charges, that's what allows water to form hydrogen bonds with other polar molecules and ions.
Right. Effectively surrounding and dissolving them.
Exactly. It's all about those charges and attractions.
Okay. Next up. What's the difference between a strong acid and a weak acid?
Oh.
Think about how they dissociate.
All right. So a strong acid, like hydrochloric acid, HCl, it completely ionizes in solution, donating all its protons.
Right.
A weak acid, though, like acetic acid, it only partially ionizes.
Right.
Some of the molecules stay together, they don't dissociate.
Okay.
And the extent of that dissociation, that's reflected in its Ka value, its dissociation constant.
Okay. And the Henderson-Hasselbalch equation, how does that help us understand buffer systems and how well they work?
So the Henderson-Hasselbalch equation, it links the pH of a solution to the pKa of the buffer and the ratio of those concentrations, the weak acid and its conjugate base.
Right.
It's really useful. We can predict the pH of a buffered solution and figure out the best range for that buffer system to work in.
Right, when it's most effective.
Exactly. Remember, a buffer works best when the pH is close to its pKa.
Now, on to the bicarbonate buffer system.
Yeah.
Why is it so important for keeping our blood pH stable?
The bicarbonate buffer system, it's like our first line of defense against changes in blood pH. It's that dynamic equilibrium involving carbon dioxide, carbonic acid, and bicarbonate.
And don't forget that carbonic anhydrase is involved, too.
Oh, absolutely. That enzyme is key.
It is.
This whole system, it's tied to our breathing.
Okay.
So we can adjust our blood pH just by changing how fast we breathe.
Right. Wow, that's pretty neat.
It is.
Okay, let's talk about those intracellular buffers now.
All right.
What are the main ones working inside our cells?
So inside the cells, phosphate and proteins are our buffers.
Okay.
Phosphate can exist in different ionic forms, so it can accept or donate protons as needed.
Okay.
And proteins, they've got all those different amino acid side chains, so they can buffer too.
So they provide a wide range of buffering capacity.
They do. They keep that intracellular pH nice and stable.
Okay, onto the kidneys now. How do they help regulate pH?
So the kidneys, they're really important for that long-term pH balance.
Okay.
They get rid of extra acid, mostly as ammonium ions, NH4+.
Okay.
And they also reabsorb bicarbonate.
To keep everything balanced.
Exactly. Maintaining that delicate equilibrium.
Okay, what's so unique about hydrochloric acid in the body?
Well, hydrochloric acid, or HCl, it's found in the stomach, and it's super strong.
Right.
It's essential for breaking down proteins. But it can also be really corrosive to other tissues.
Yeah, it's a powerful substance.
It is. But luckily, the small intestine neutralizes it.
With bicarbonate.
Exactly. Yeah. That protects the lining of the intestine from damage.
Okay, next question. What is osmolality, and why do doctors need to understand it? Think about fluid shifts.
Osmolality is all about measuring solute concentration.
Okay.
It basically tells us which way water will move across those semi-permeable membranes like the ones around our cells.
Right.
Doctors need to understand this.
Yeah.
Because it helps them manage things like dehydration, edema, and electrolyte imbalances.
Right, so it's about keeping those fluids in the right places.
Exactly. It's all about balance.
All right, let's talk about isomers. How can two molecules with the same formula have different effects in the body?
So isomers, they might have the same formula, but their atoms are arranged differently.
Okay.
And that can make a big difference in how they interact with biological systems.
Right, like those mirror image ones, the enantiomers.
Yes. Enantiomers are a great example. One enantiomer of a drug might work perfectly, but its mirror image might be useless or even dangerous.
Wow, that's wild.
It is. Structure really matters.
It does. Okay, now for some energetics. What is Gibbs free energy, and why is it so important in biochemistry?
So Gibbs free energy, we call it G.
Yeah.
It basically tells us if a reaction will happen on its own or not.
Spontaneously or not?
Exactly. It combines enthalpy, which is H.
Okay.
That's the heat content of the system.
Right.
And entropy, S, which is about disorder.
Okay.
If the change in Gibbs free energy, delta G, is negative, the reaction is spontaneous.
Okay.
It'll happen.
Right.
Okay, last question. How do enzymes affect the energetics of a reaction? Do they change that overall Gibbs free energy? Great question. Enzymes, they're catalysts. They speed things up.
Right.
But they don't actually change the overall Gibbs free energy.
So they don't create energy?
No, they just make it easier for the reaction to happen.
So they lower the activation energy.
Wait. Exactly. That's the energy needed to get things going.
It's like they provide a shortcut.
You could say that. They make the reaction happen faster.
Amazing. Well, I think we've covered a lot of ground today.
We have, from the basic building blocks to those intricate metabolic pathways.
It's been a fantastic deep dive.
It has.
And I hope everyone listening feels a little more prepared for those biochem exams.
I hope so too. Just remember, biochemistry is about understanding those elegant principles that govern the chemistry of life, not just memorizing facts.
Right, it's about seeing the big picture.
Exactly. So keep asking questions, keep exploring, and you'll do great.
That's all the time we have for today.
Okay.
Thanks for joining us on the deep dive. And best of luck on those exams.
You got this.